Yeah. Yeah, please settle in. Quick, quick. Don't take too long moving a seat. You come to the side of. Yes. Yeah, we can start. Great. Actually, according to my time, it's still at 2 minutes. So, but if everybody's here, they're not waiting for anyone else. Yes. Okay. So, good morning, everyone. And good to have reasonably full hall for today's mini symposium. So, am I audible? Fine. So, every year we have for the Journal of Genetics an editorial board meeting. And for the editorial board meeting, traditionally it used to be an offline thing. Nowadays it's somewhat more hybrid. But we have a bunch of editors who do some uh, for the meeting, and we thought that that would be a good opportunity for not only each other, right, for the editors, fellow editors to know what the area of expertise of each of our fellow editors is, but we can also sort of take advantage of the fact that they come prepared to talk about their work to invite students, from local colleges and others who are interested. And so this idea of a mini symposium developed. We are very thankful to the staff of the academy for helping us put this together. In fact, it's their initiative, and I would like to formally thank Mr. Mahesh Chandra and Mr. Sumesh and Mr. Rakesh, and also the staff of the editorial office of the Journal of Genetics, Ms. Gracie, who was outside, and Mr. Arun. And there are a whole bunch of other people yeah, whose names I won't take, to make it all possible, right down to details like uh, connecting us with online. One of the speakers is going to join us from Canada. And one of the reasons for keeping to time is he's 8 hours behind us. So right now it is about 2:30 in the morning for him. Right? And at some point he will wake up and presumably join us and deliver his presentation. So it's against this background that I wanted to tell you people. And each talk is meant to be for about 30 minutes. Right? And the first talk is one that I'll be giving. It just turned out that at the time we wanted to finalize the program, we had 3 people who had agreed to speak, and the fourth one was still not sort of on the horizon. And I decided that rather than try to get one more speaker, I would step forward and speak on the topic that is up here, which is DNA profiling and ethics. Right? So, my name is Vikas Kaswekar, and I'm a PINSA honorary scientist. That means I'm actually retired from the bench. I'm no longer an active scientist at the bench. But I have a connection with the, continue to hold my connection with the CDFD, that is the Center for DNA Fingerprinting and Diagnostics. Uh, and my when I decided to choose a topic to speak on, and I didn't want it to be about lab-based science because I stopped doing that. I wanted it to be something that I felt that I'm still engaged with, and the area that I'll be talking about is something called DNA profiling and ethics. It's a very unusual combination of topics, and I'll show you why. Right? So oh, this takes a second. All right. So this slide shows a picture that I downloaded from the web, and it's a very sort of nice picture I find because there are 2 twin sisters, identical twin sisters who are married to 2 identical twin brothers. Okay, and it sort of is a quick way of telling you why the twin brothers look so much like each other and why the twin sisters look so much like each other, right? And the basic thing is that they look so much like each other because by and large they share their DNA, right? And where is the DNA? The DNA is within the cells, right? Each of them has a few trillion cells, right? And a few trillion cells may sound like a large number, but it doesn't take too many cell divisions to reach a few trillion. Right? If you have one cell division, you get from one cell, you get 2 cells. 2 cell divisions, you go from 2 to 4 cells. 3 cell divisions, beg your pardon? There may be, yeah, there may be more bacteria. I'm just talking even about the number of cell divisions that you need to go from one cell to a trillion cells. Right? So, having said that, does anyone have a guess as to how many cell divisions in sequence you will need to go from one to a trillion? Okay, I thought no one has been thinking about numbers for a long time. From 1 to 1,000, you take 10 cell divisions. Okay, you go. If you do 10 cell divisions in sequence, you'll get 1024. So if you do 20 cell divisions in sequence, you'll get 1024 into 1024. That's for simplicity, call that 1 million. And if you do 40, you'll come to 1 million. Right? So it's only less than 50 cell divisions from the time each of them was a single cell. Right? And actually, it is one for identical twins is one more cell division. They actually came from the same zygote, and basically they so they had one extra cell division uh, each when since the time they separated. Okay, so within the cell, inside the nucleus, there are these bodies called chromosomes, which are visible like this only during cell division. They are called they condense for mitosis. And within each chromosome, you have a DNA module. And the DNA module I won't describe because I take it that most of you are life sciences students. Even if you don't know what the structure of DNA is, you know where to look for it. So I won't spend time telling you about that. But what I'm going to tell you is that the DNA sequences, right, or the DNA sequence on a molecule can be pretty long. Right? It can be of the order of 3 billion base pairs, right, in the human genome. And there's an interesting range in the rate at which DNA changes. You may think that these two guys look more similar to each other than these two girls, right? Maybe. I mean, I've been sort of wondering about that. It could be because since the time they sort of separated as two cells, they have accumulated different numbers of small numbers of mutations. Right? It could be that, or it could be something else. We don't necessarily understand everything that contributes to the development of an individual. Could be, yeah. So what we have is that there are parts of our DNA sequence that haven't really changed. They are like, older than the rocks. And when I say older than the rocks, I mean it. Because there are sequences that are identical in humans and rats, yeah, which both share a common ancestor about 80 million years ago. Okay? They are called ultra-conserved sequences. They are not too long. They are about 200 base pairs. Yeah, that's the definition of an ultra-conserved sequence. 200 base pairs without a single difference. So if you were to read that sequence from a human and you read it from a mouse or a rat, you won't be able to tell which one you are reading because they are completely identical for 200 base pairs. There's nothing in biology that we understand that currently explains why the sequence should not have accumulated any mutations. So that's one end of it, as solid as a rock between human and mouse 80 million base pairs, million years ago. At the other end of the spectrum, you have sequences that actually differ between father and son. The reason I say father and son and not say parent and child is because they have been demonstrated on the Y chromosome. And you can compare the Y chromosome of a boy and his father. And in one generation, actually, there are some regions of the Y chromosomal DNA which have actually undergone changes within a 25-year period. Right? So, on the one hand, you have changes that happen in parts of your DNA within 25 years. On the other hand, you have things, other parts of the DNA that are not changed for 80 million years. And between these two extremes, right, there are sequences of various kinds. And there is one category of sequences that are very useful, which is used for doing DNA profiling. And those are the simple tandem repeats. Right? So these are the simple tandem repeats. There is a simple, say, 3 or 4 base sequence repeated. In this case, there are 8 repeats, here there are 7 repeats, here there are 9 repeats, just for illustration. And you can actually determine, the repeat size because you can amplify that fragment using blanking unique sequences as primers in the PCR. Yeah, again, assume all of you know what PCR is like a primer. So basically, by using the same set of primers, if you were to amplify from this DNA molecule, you get a size corresponding to 8 repeats. From this DNA molecule, you get a size corresponding to 7 repeats, and from here you get for 9 repeats. Right? So you can actually now resolve these differences in a capillary column, right? By running it along a capillary column, electrophoresis in a capillary column. That's done over here for a set of markers, and in every marker you'll find that there are 2 peaks. Right? That's because there are 2. Just give me a second. There are 2 peaks because there is 2 source DNA molecules, one on each chromosome. Right? There is one chromosome that we have inherited from our father, one chromosome we have inherited from the mother, and therefore the sequence that maybe the paternally inherited chromosome had 8 repeats and the maternally inherited one had 9 repeats. Therefore, you can resolve them for every such locus. So a bunch of loci have been picked up. I won't go into details of what these other lines are there. They are the different sizes that are known. And I won't tell you much about why the different colors, because we do it in a multiplex kind of format, right? So we can get a DNA profile. And these are the different markers that are commonly used. They are one set of markers. We could have had hundreds of other loci. Because in the genome there are many STR loci, simple tandem repeats, right? And this set was chosen because they are on different chromosomes, and the FBI has been using it, and once they started, the rest of the world follows using the same parameters. Right? But we can, if we want to be contrary, we can choose another set of parameters. There are hundreds of sets we can create actually. So this is at this point in the slide. Yeah, mine. How stable are these? Yes, yes. So they have been chosen because they are stable enough. Okay, so I told you about the Y chromosome repeats that were found. That was a study done by Valentine and co-workers, where they actually looked for simple tandem repeats on the Y chromosome that did show differences from father to son. Okay, so those are called rapidly mutating YSDRs, RM YSDRs, right? And because they were on the Y chromosome was easiest to check, they have been defined there. But there's no reason known to us why they can't exist on the other chromosomes. They have not been identified from there as to the best of my knowledge. Right? So rapidly mutating STRs would be there on the autosomes as well, but they would not be very useful for us because if they are mutating at such a rapid rate, then you can't actually say that this person is the father of this person or this person is the brother of this person because the repeats keep changing too rapidly. Okay? It's like changing of clothes. Today you are wearing a red shirt, tomorrow you are wearing a yellow shirt. You can't say that on which day you are wearing which shirt. So I don't know if this analogy has made sense. Yeah, but these are also repeats. So we don't understand why some repeats are so stable and why some others are not. Okay, there's a lot we don't understand. We don't understand why golden base pairs are identical between humans and rats. Okay, in fact, there have been the standard experiments have been done. When mutations have been put, there is no function found. So basically, these are mysteries, right? They are mysteries in the genome. So as I speak to you, despite the fact that very brilliant evolutionary biologists and biochemists and molecular biologists have been now pondering these questions, no one has an answer for either why some segments of the genome have not changed for 80 million years, and no one has an answer for why some segments of the genome change within 20 years. Okay, so these are the two extremes that are mystery, but we are dealing with something that has been chosen for not changing so rapidly. But in CDFD, we have been doing hundreds of profiles, and we do occasionally see mutation. One of the things I told my colleagues in CDFD is to see, whether mutation frequencies are occurring at higher rates in certain populations versus others. So we have all this database, but frankly, they don't do it. I've been saying this for the last 10 years. No one's done it, and they are probably wondering what they were so keen on. Why didn't you do it when you were active? Right? So that's I will have an explanation for that. But so for coming back to this, right? We have for each locus there are 2 peaks. You understand why there are 2 peaks, and let's not get into how we resolve these peaks and so on. Just take it from me that it can be done. It's done routinely. That's how we get a DNA profile. And what we do next is we make a table, right? And this table is used for our DNA profiling service that the CDFD does. When the courts or the police or investigating agencies ask us to do it. This was an actual case, right? So here is the seal of CDFD. Here is the title saying set about DNA fingerprinting and diagnostics. And this is a case where this lady, right, had been presumably sexually assaulted, right? And this is the DNA profile of the baby from that assault. And she had said that this individual, some man, he has a Y chromosome, was responsible for that sexual assault, right? So we got this case from the courts or the police, and we made the profiles, and we wanted to establish whether this is true. So for the lady, for every we call her henceforth the victim. Yeah, in any sexual assault case and wrong, the way we deal with it is we call that person the victim, and that's the victim's baby, right? In the middle. So you can see the victim had the markers 12 and 13, and 13 came into the baby. And there was another 13 that came into the baby, but the father, alleged father, does not have a 13, right? But at a different locals, you have say 14 and 16, and in the mother, and 14 and 16 in the baby, and you have 14 here. So it could be that this 14 came from the father, right? And this 16 came from the mother. So at that locus, it is possible. But when you look at the whole thing, we have marked out in red the loci that cannot be accounted for as coming from him. Okay? So based on that, we can actually say that he is not the perpetrator of the assault that led to the conception of the baby, right? We can say that. Are you all with me? Okay, so basically this was the report was prepared, duly signed, and it would have been the end of the matter. We would have said that he is not the perpetrator, he is not the father. But this is now where we come to there is the standard technical work that many people without too much advanced training can do. And there is something which where a scientist who has spent his life wondering about science steps in and sees that there could be something more there. And when I saw these profiles, I basically highlighted, right, some of them. What are the ones I have highlighted? The ones I have highlighted is where the same allele is present on both copies. That means you get, instead of getting two peaks, you get one double peak, one taller peak. Okay. So here we have 13, 13, 13, 31.2, 31.2, 10, 10, 12, 12, 6, 6, and here 12, 12, 12, 12, 12 across. Right. So I just highlighted this. Okay. Now what does result of the highlighting tell me? That in the baby, right, whose father this person is not, there are actually very large number of homozygous loci. Okay, so this is sort of gets me thinking. Too many homozygous loci in the baby, right, from a sexual assault. The alleged perpetrator of the assault, yeah, as presumably accused by the victim, is not the father, right? Something's fishy. So this is where, once you have the report, but someone is thinking about it, begins to wonder that there's too many loci that are homozygous. And she probably knows who the assaulter was. Okay? Because, I mean, with all said and done, yeah, at a fairly early age, yeah, the unfortunate thing is women who are sexually assaulted get to know who the person is who has done it. So why is she not sort of pointed out the right person, the real one? Why has she pointed out someone who is not? These are thoughts that go through one's head. I'm a molecular biologist, geneticist, and all. These are not things we are trained to think about, but one has time on one's hands, one starts thinking about that. Okay. So one possibility that comes to mind is this: could it be a case of incest? Okay, where incest cases have been dealt with in the CDFD. Yeah, father rapes his daughter, brother rapes his sister, and we have had to deal with such cases as well. Right? But here the question comes up: why does it come up? Because when you have father-daughter incest, right? So here is the father, squares or rectangles are the males, circles are the females. So this person, right, has a daughter, and he has had sex with his daughter, and basically this is the baby from that divorce. And when mating has occurred between those relatives, we put a double line here in Pedigree. And if that happens, then the probability of any gene becoming homozygous can be very readily computed as 1/4. Because if one allele has come from the father, what's the probability that the same allele has come from the mother? Mother in this case is his own daughter. Right? The probability that, say, one allele has come from the father into the baby, the same allele going into his daughter is half, and that allele being sent down into the baby is another half. So the probability is 1/4, right? That means the probability of getting homozygosity, right, for a particular allele is 1/4, which translates to 1/4 of the loci would be homozygous, right? This is actually more than 1/4. I will be easy to count. It's actually more than half. So basically, this comes to mind. So, now this is an additional input which someone who doesn't know about what happens when there's mating between two related individuals, right, may overlook. In fact, this was overlooked by all the people who were in the DNA profiling lab, right? They come in the morning like everybody else, do their job and make a report and so on and so forth. And they found that the report would simply say that the alleged father is not the real father, right? And they would have been fine, nothing to complain over there. But given that I know this, I know that your father, daughter, or brother, sister is 1/4. That would be good enough reason that a person like me who knows this, when he sees this, right? Because I took that trouble to actually see which is homozygous or how many are homozygous. Right? What should I do now? Right? To the investigating officer. This is where the ethics comes. Right? The police and the courts and so on, they have many cases. They would ideally like you to answer the question they put to you. Is he the father or not? And we say he is not the father. Case closed, go to the next case. But we know that she knows who the real father might be. This data here suggests that the real father might be a first degree relative, and therefore he is not in a position to say so. What should I do? First of all, I have to explain to my own colleagues that this is a possibility. Right? Is this the only possibility? No. There are alternative possibilities also that can explain it. One is called endogamy. Right? In many populations, especially in India, they marry within the same group, endogamous group. Right? So they are not necessarily related, but over generations they have been marrying only in the same group. Ayers marry Ayers, Iyengars marry Iyengars, and I believe hell breaks loose if there is any cross marriage. Right? So these kinds of things happen. You can see it in the paper, right? And we also have many communities where there is uncle niece marriage, right? So, and that is considered legitimate. There is no problem. In fact, just this morning somebody was telling me that in the family there is a concept of uncle niece marriage, right? So, and there are first cousin marriages allowed in one direction and not another. So, there are marriage practices in different communities. So, there is a range between what is really taboo in pretty much all communities, right, versus what is not taboo. So we have to, and there is endogamy for many generations. So we have to be mindful that we don't simply say that arrest the father or the brother of this woman. Right? This is where the boundary, this is where the ethics come. Right? Now, philosophers and ethicians and so on, they think about big problems. They don't think about a small problem like this. What should we do? Right? Geneticists, yeah, and DNA profiling technicians and so on do a competent job. They'll make this table. Who should be doing the thinking of looking at the table? It's not something for which we are, it's not part of the job description, right? And yet, I think all of you will agree that this is something that we have to flag. I brought this up in a course I teach these days on research and publication ethics. So just ethics in general, right? And because one has to teach a course in ethics, it has to be done in a way that you can skip over topics quickly. And for that, I make multiple choice questions. It's not easy to make multiple choice questions in ethical problems, but if you make them, they allow you to sort of sample different kinds of problems. One of the questions I put was using this problem. What should the DNA examiner do? One choice I put in, some 4 or 5 choices. One choice I said that it's clear that the victim lied when she said that this individual was the father. Right? So the police should continue interrogating her until she really comes up with the truth. And I was actually shocked to find that some of the students touched that as the right answer. They felt that this should be done. So it goes to show that even out in society, right? How many of you, by the way, think that the police should continue? I kind of gave away the answer, but how many of you think that the police should continue interrogating this woman, the victim, until she reveals who the real guy is? Right? In this, I think most of you know that I consider that that's the wrong answer, and I have not said it. Some of you would have raised your hand and said that that is wrong. Yeah, please raise it.
For the defense counsel,
What?
point out this woman's line, you must show that this line is innocent. Not simply by saying that this is for the father. Saying that this woman's line in the form has to be doubly, it's double value for her.
No. As far as DNA is concerned, this person is not the.
Defense.
Yeah.
Yeah.
No. Defense lawyer will be fine with this, and this guy will be let off.
No, it will be like additional proof that he's lying. So you can't.
Correct. So now this is where we want to discuss. So basically, these are not things that you and I have expertise in, right? I have expertise in computing these numbers, right? You probably have expertise in running those capillary columns and so on. But who amongst us has the expertise to be able to say what we should be doing in in our report? So basically, there, firstly, you'll be amazed by how ignorant judges and police are of even things like DNA. Okay, so I have interacted with them, talked with them, and then it is pretty scary. So incompetence is pretty rife. Yeah, in the world. Yeah. So and I'm sure I'm talking about an area in which now I have some expertise. I'm sure that somebody will be able to point out that there are several things that I'm pretty incompetent at. I know now. But in this, what should we be doing? So basically, I first raised this as a part of this course, and then I found that almost nobody had really thought about these things in a place like CDLB, right? Where we deal with this. Nobody has thought. Nobody has thought about it academically. Okay? So what should we be doing? I had a just a stopgap kind of measure. I said that you should tell the investigating officer that look at the first degree relevant, right? People have been doing that. If you go to cold cases that have been solved here, particularly in the United States and so on, they do all kinds of things like that, right? It's easy to do. You get some trash outside, chewing gum or something, you get the DNA from that. If you know that this is gum that was spat out by the father, or palm spit or something like that, one can do it without arresting the person and so on, and then one can follow it up. But should that be done? There are privacy individuals who say that that's infringing on the privacy of a person. Right? Where does privacy rights come, and where does the woman's victims' rights come? So I'm not going to pretend that I have the answer, and anyway I've run out of my time. So I'm going to leave you with this. Right? DNA profiling and ethics. Something to think about. right? So this is, I'll stop here because I've been very, very particular about everybody else being on time and let me talk about my thoughts. So I'm going to stop here. But if there is one question, since I'm the chair of my own session, I will give me the prerogative to answer one question. If anybody has one question. Other than that, over tea and so on, and lunch, you're free to come and ask questions. Question by anyone other than mine, please. Nothing. He's done this quota. Anybody? Especially among the students. In fact, you. Yeah. You're passing off as a student. You're a student. Yeah. Okay.
Yes. Do you think that apart from this ability, more genomes evidence
Not for this exclusion, this is adequate.
that is not
Yeah. As far as exclusion is concerned, it's adequate. In fact, I told you if there are parts of the genome that are rapidly mutating, they will make you falsely eliminate, exclude someone. Right? So basically, we have to be. By the way, I think I forgot to tell Sumesh to start. You know, we started. I hope the that's okay. We can start with the next. So getting the whole genome does not address it. This is what is done. It's in the court cases. They have gone through hundreds of court cases. It's accepted around the world. Yeah.
There is another issue. Very good. This is the.
Yeah. So that's so we depend on the chain of custody. Okay, we sort of verify that when the DNA or the samples were sent to us, yeah, they were brought to us by two police officers. Their names, rank, numbers are taken. They were received by three people in CPFD. Some of them were not involved in the DNA profiling at all. It's done blind. All those SOPs are there, which is why it can stand the test of being in a court. The obvious things have been done, to make sure that. Yeah, I mean, a police can do all kinds of things. I mean, there are people being shot and killed, figure on in UP and on the road. Yeah, that's the reality. So, okay, I'm going to stop here and invite the next speaker, Upendra. He's going to introduce himself and keep that.
Yeah, no. I got lots of data. Then I also realized that many of you are students not doing research. So what I will do is that the most of the data I will skip, right? I will just go through it. You don't have to really know the data. But what is important is the techniqueness, is that it is complicated title, but take it from me, what I'm trying to say today, when take home message is that you can utilize the model system to not for the drug development, right? Okay. Um, and lots of collaborations are important because I have this work would have not been possible without the help from my colleagues from the other institutes, right? Okay. And most importantly, resource sharing, which we need it in India at the moment because the fundings are very scarce. Yeah. So my group worked extensively on the model organism, particularly these two animals, Drosophila and the zebrafish, as a vertebral model, and this as a single gene mutation model. Okay. So we worked on extensively on muscle development, neurological disorders, muscle diseases, and we also work on the immunity using this model organism. And recently, we have been extensively working on health span, means how you can die without depending on the other people, right? So, our quality of the life, as you know that, why we got into particularly in the epilepsy is that in Bangalore itself, right, in India, 9 in 1,000 people suffer from epilepsy, which is very, very high. And particularly in the outskirts of Bangalore, if you look at it, that number can go from 9 to 16 per thousand. Okay? So it is very high. The other thing is that the most of the available drugs for the epilepsy, if you use it for the long time, then they can develop mobility disorders called the dyskinesia, which is the major problem. And the second thing is that the 30% of the patients actually they develop what we call the drug-resistant epilepsies. Okay, the whatever drug available in the market, you can't use them for these patients. All right. So I got excited because I always wanted to work on something where we can cure people, right? Okay. And the other thing was that somewhere where our approach is different, right? Unlike the allopathy and the drug screening, we wanted to utilize the knowledge that we had it. And for example, particularly, I'm fascinated by Ayurveda, and so we wanted to take the lesson from the Ayurveda and see if some of the claims in the Ayurveda can be scientifically, proved, or can we provide empirical evidences. That's right. So that the epilepsy can happen to anyone and anytime, and these are the what we call as the unprovoked seizures. Okay, and if you look at it, most of the doctors they can use ECG or EEG. The neurons should be keeping on firing at the abnormally, and depending on the where the brain is happening, where the defect is, you can actually do surgery. If it is very close to the thing, that is what the layman's most of the doctors they do it. If it is not going to impact the life per se, they can remove those small portion of the brain, and then most of them get cured. Okay, but many times, for example, in the focal place, it's very difficult to remove a chunk of the brain, right? That is where the drug comes in play, right? And depending on where the brain is, what kind of things behavior they with. I mean, the seizure can be multiple types. For example, for the small myoclonic or the tonic, repeated once, and things like that, and both together. Okay, so the epilepsy, there are many clinically heterogeneous groups. So I'm not a clinician, so what I know at the molecular level is that it happens because of the abnormal firing, and that is what we should think about. Okay. So far, the problem you have is that the epilepsy can develop from, for example, genetic. I mean, only around say 5% of the epilepsy you have a genetic origin, but most of the other things are either from the head injury or infection and many things, right? Okay. Um, so the treatment available, as I told you, many develop non-responsive here, and out of the 18 available in the market, I mean, four drugs are given to the this epilepsy patients, and even these four, if you use it for the longer time, then you can develop. So we wanted to look for the novel way of looking it, particularly deriving from the plant, as I told you. The other thing, let's not worry about too much. The other thing what we realize is that most of these available drug target only the last portions. Yeah, they never once the symptoms start. That is where the most of the patients go to the hospital, and they target only the relieving the phenotype. Okay, not initial stages. These things, right? So we wanted to study the etiology of all these epilepsy conditions, which you can do very nicely using the model organisms, and at what stage if you intervene, maybe it will work, right? So we, as I told you, we were fascinated by. Let's not worry about it. So the defective here and defect how we can control this was the major problem for us, because there is no time. So I will just pass all most of these things. So what we were trying to do is that there's something which happened, right? Uh, so we wanted to use, particularly we have shown that the pomegranate is a good source, and in the Ayurveda text it is well written that the pomegranate actually, if you take it normally, it is a rasayana, okay, Ayurveda rasayana. So on its own, it acts as a cyanide, revitalize all your body. And what we have shown in 2014 is that pomegranate almost improve your body, right? Okay. Only thing is that you should take for 3 months, then you take a break in between, right? Any Ayurveda drugs should not be taken continuously, right? Even if it is. The good thing about the pomegranate is that so far people have shown that allergy terminals they can actually the product which is available in pomegranate, right? Okay, you find it one of the polyphenol is the allergy terminals, and it works fantastic. But we were skeptical because many times what happened is that the Iraqi channels are broken down to urethane A, B, C, D. These are the kind of things, smaller polyphenols, right? Okay. So we wanted to check that this could work, right? And the other thing is that the Iraqi channels are converted to urethane by firmicutes in the our blood, right? Okay. I will come back to you at the end that why we should work on urethane rather than take pomegranate or the energy. Okay. So we wanted to check if the already people have shown that if you look at urethane A works for fantastic for the neurodegenerative diseases already. My friend Orex, they have a big company on urethane A. And they have shown benefit for the muscles and things also. Okay, so we wanted to check that whether the urethane is the one which works, right? So in a nutshell, what we have done is that we have used Drosophila as the initial screening thing, right? Okay, because Drosophila we can induce chemically, which I will show you the data. Then we have multiple genetic epilepsy models we have generated in the lab, so we can put urolithin in the all these mutants. I mean, how many of the mutations actually recover the phenotype, right? In that way, you can tell that whether this compound works for the what kind of or the channel mutations or let's say other genetic mutations, right? Okay, that is what we tried. Once we have it in Drosophila very fast, then we validate it as a preclinical thing in mouse model, and we try to identify the mechanism. Okay. So picotoxin is a antagonist inhibitor of the GABA neurotransmitter. GABA is the inhibitors. Okay, so you can induce epilepsy by using with the PTZ or the picotoxin also PTX, right? So we use it. Let's not get into the things. You feed it the picotoxins. Okay, it doesn't allow the GABA to inhibit so that the neurons will get excited. That's the whole idea. Yeah. So you can just do this measurement very easily. You don't need sophisticated technology. You just observe it. Nowadays, you have the iPhone. Actually, we don't even use a microscope. Good iPhone, you can observe how these larvae are crawling. One example is here. So, okay. So you can see that once you treat with a chemical, they don't move, the others will move around, right? And the same chemical with the urolithin with A now will start moving. Okay, this is the data. And what happens, right, at the neuronal level? So that is the good thing about using model organism. So you can look at what happened in all this neurotransmitter region when this is called buttons. Buttons are the neuromuscular junctions. Okay, so you can measure that. All this experiment you can just do it in Drosophila, right? Okay, very nicely. So you can see that the chemical treated ones, right? Here you can see the recovery when you put the urine in. As I said, the same thing, whatever we do it with the chemical induced epilepsy, let's not worry about all these things, right? It will take lots of time, so let me just go through it. So what we have shown here is that whatever you get it with the chemical, actually, it can be recovered by using with the urine. That's don't worry about the details of the data, right? And this has been found, right? We measure this frequency of what we call as the excitational junctional currents and things like that, electrophysiology. All right? This is what the drug epilepsy happens, and we did the same thing with the say genetic induced epilepsy. Right? Genetic models you can use it where we have a mutation called the bang sensitive. Right, this is very sensitive to the like if you just bang the fly, they start showing the epilepsy, and this is because of the mutation in the one of the mitochondrial channel. Okay. Right. So you can see that with the band sensitive, the defect is there with the urolithin, actually you will start recovering it. And the same thing again can be shown by electrical signatures and quantities. Okay. What we have done, so what we have shown is that the increase with the chemical or the mutations where there is an increased release of the neurotransmitters, actually we were able to inhibit activations, right, with the urolithin. That's all. Right? Now, question comes is that whether we are moving from here to this reversing. That's all. So what we need to do is that we need to see is that how this urolithin is working, right? So for that we went to the mouse model, preclinical, right? Yeah. So the first thing what we did was the mouse brain slice. If it is working as the extracellular, so what we do is the epilepsy, we will put it from the outside urolithin, right, in the brain slice. If it is actually going to recover the synapse, then what happened? Not the electrophysiological, you can measure this. If it is going to recover, it means that maybe it will work extracellularly. But we didn't find any difference in this. It means that it has to be intracellular. Right? In that condition, what we have done is that how urolithin is working. So that we give them chronic urolithin. Okay. By the way, urolithin is not toxic to the animal. Right? Okay. To say few grams, actually, you can take it, and it is not toxic. So we gave it, injected the urolithin to the mice. Then we look at how long it takes for the urolithin to reach the brain. Okay. Again, to cut short the whole thing, as soon as you put it within say 30 minutes, you can see urolithin in the blood of the serum of the mice, and within 45 minutes, actually, it will reach the brain. So it can pass through the blood-brain barrier easily because it's a very small, right? It's very important. The other thing what we noticed was that actually urolithin get modified, right, in the serum, but as soon as it reaches the brain, again it comes to pure urine. That's fantastic, and we don't know the actual mechanism how it works. Okay. This is the data from that whole thing. I will not show the whole MS and the LCMC and all those things what we have done. See, we are not going to show you. But what we have done is that we use the mouse model, as I told you, you induce the chemically and you just start checking that at the what stage the urolithin has to be given so that the mouse recovers from this mice recover from the epilepsy. And that is what we have shown is that you can see it, right? Not work, but I'm just cutting down, right? Okay. So what you can see is that we were able to see the recovery of the seizures in the almost half of the animals, right? Okay. So urolithin actually can stop the epilepsy, and that is what we have shown. Now the question comes is that how it is doing it. As I told you, the good thing about having a mouse is that you can do fantastic, just like in the Drosophila, you can do single neuron measurement. You can do it, right? Okay, by using a PET scan, and that is what I have been working with my colleague James Clement in the GNCSR, and we were able to show. Let's not worry about all these methods, okay? And these are we are calling about the kind of spontaneous firing, means without evoked firing. Most of the epilepsy patients, they have this what we call the miniature evoke firing. Okay? So small firing, and that actually gives rise to all this thing. And you can see that, let's not worry about the whole thing, but you can see that as you have it with the chemical, these urolithin treated ones actually recover. So that miniature firing of the neuron, remember that in the long run, actually they can generate to your fatigue in the muscles, okay? And that fatigueness can cause multiple things, cell death, even in the neurons or the muscles, it can generate it. But here we were able to recover it, right? So far the story is that the urolithin works fantastic, right, for the epilepsy patients. And now the question comes is that let's not worry, this is too complicated. Right? So we look at some of these cells which are post-synaptic and pre-synaptic. So you can check it, and some of the things which we have looked are these are very important. Chemically induced one, PSD 95 is the post-synaptic protein which is required for these contractions. And you can see that in the chemical induced one it is less, but it is recovered in the urolithin platforms, right? Similarly, we have looked at the multiple downstream targets, so let's not worry about it. And the only thing what we have to see is that here it is able to recover it, right? Now, let's look at the mechanism. The thing is that the urolithin, if it is working, how it is working, and the things. That is the good thing about, as I say, collaborations, right? I'm not a chemistry person, and I can't do it. Right? So we talk to my colleague in ISC. That is the good thing about working in the ISC. You can just approach anybody, right? Particularly if you haven't harmed them, you can collaborate with anybody, and I haven't done it, right? So I approach these people, and we are the only 8 labs in the world to do this kind of work, by the way, right? Okay, so, huh? Yeah, so what we did was the urolithin is a fantastic compound, so that we can link from the non-functional group. So that we provided a linker so that it can be formed as a bit, right? Then because we have the brain sample to pass through this bit and trap the protein which interact with this. And this is exciting because not for this person, it would have not been possible. Okay, this boy, Sambit, PhD student of Tilaga, he took it as his own project, right? Because we collaborated with another PI chemistry, and his student they could not do it. But for Sambit, he said, okay, let me do it, right? He took 8 months to do all these things, and luckily for us, we were able to pull down, right? And these are the linker how we get it. And this is what the fantastic about, right? So we were able to pull down this protein, and this is nothing but the voltage gated and selected we did one, right? Okay, we did one actually is already shown in the sheet that actually it becomes abnormal, and the one our actually urolithin is doing is the binding to this protein, right? Both the VDEC1, we believe in the VDEC1 because the VDEC1 we were able to pull down the tubulin actually which form dimer with the VDEC1. Okay, so in the nutshell, we were able to identify the target of the urolithin. So the question is that if it is working through the VDEC1, then we should be able to recapitulate whatever the VDEC1 does. And we have done all these experiments, and I'm not going to show you this data, okay? Because all these things will take lots of time, but the one I'm going to show you is that if it is going through the VDEC1, that is the good thing about coming back again to the fly genetics. So that we were able to knock down the VDEC1. Let's not worry about this chemical, all the data we have done, RNA, and everything. We were able to pick up the genes, which are the direct targets, and we have modeled it. But this is important. So that you knock it down, VDEC1, we should be able to recapitulate what urolithin was doing, because urolithin was inhibiting the VDEC1, right? So we did that, and you can see that when we broke down the VDEC one, actually, you are able to recover this, right? And the good thing about the, again, our belief is that because VDEC one, we were using the RNAi thing, so that it will not knock out completely the VDEC one, right? So the VDEC one with the urolithin actually it gave a synergetic recovery, much better than the VDEC one knockdown alone. Okay, so urolithin works, right? So this is really exciting for us. We have just put this for the provisional patent in India, and we because nobody has done this, and we have put both the US and the European patent for the how the urolithin is, will be able to cure the epilepsy because it works fantastic. Unlike the many of the available allopathic drugs, it works on the one of the most important within channel protein, and it recovered it. And this would have not been possible without this student just not know about it because it worked irrespective of the epilepsy because we have used all the model organic, model epilepsy model in Drosophila, whether it is mitochondrial or channel, sodium channel, or say chemical induced and things, and urolithin works. So we are very confident that it will work for the almost all the epilepsy, different kind of epilepsy, without actually looking into the different genetic system. And so, right? Okay, that is the good thing about it. And this would have not been possible without this girl. Like that is where I feel that when you have a good student, it can make a huge difference. And when she joined, she was adamant that she wants to work on neurobiology and epilepsy, nothing more. So the epilepsy work started with her in my lab, right? Okay, and you think about it is that she got trained herself in the electrophysiology with less in the isopropyl, and she got trained in the PES clamp with Professor Sigder in the IAC, and most importantly, she has worked extensively with the James Clement in JNC area, ASR, and with like just she went from from Drosophila to mouse geneticists to almost everything, right? Okay, so that is where I'm excited about the students is that student can make a huge difference in your career, right? And I started learning epilepsy, right? Otherwise, I am a muscle person, right? Okay, I stop here. Thank you very much.
Yeah, time for one question.
Yeah, I will just tell you is that why pomegranate eating doesn't work is that I told you 40% of the our population, we don't have enough CFU or molecules in our gut, right? Those who this will convert the pomegranate like channel to urolithin. So if you take the final product, it is much better, right? Particularly if you are suffering from autism and neurological disorder, then you don't have this bacteria microbes in your gut.
Yeah. Let's have questions. Any questions?
Read
Feedback on.
yeah, read that one. Is it kind of target for many of the drugs? I've gone across two papers where which during a retinal damage, scientific retinus.
So VDEC one is a portion of here, which is particularly important for the mitochondrial function. So you can target it, but as much as I know it, people have been talking about it, but there is not even a single drug. But people have shown that VDEC one is important for the many of the physiological activity, but there is not even a single drug directly targeting the VDEC one so far, and we are the first.
So downregulation of VDAC is kind of recovery.
It's a double-edged sword because the VDEC one actually in normal, I haven't shown you the data, but in the VDEC one in the normal condition, actually urolithin activate the VDEC one, which is good. In the normal, in the abnormal conditions where VDEC one is inhibited, that is, activated much more. That is where you need to inhibit.
Yes, balance to be.
Yeah, it needs to be balanced.
Any Any other question? Yes.
So my first question is how common
One
is
question.
this?
So we may have to go to another question.
How common is this disease in India?
India is, as I said, it is 9 in 1,000 per 1,000. It is already data is there. Epilepsy is 9 in 1,000 per people. It means 9 in 1,000 is very, very frequent. One of the most frequent.
Approaching 1%. He said 9 to 16 is
16 in outskirts of Bangalore. We don't know why, but the data suggests that up to 16 in the outskirts of Bangalore.
And is there no side effects of this chemical?
Yeah. But urolithin is a something which you are eating with a pomegranate or many fruits.
While directly injected.
Directly injected, my colleague Orex, they have shown that up to 5 grams you take it per day, it doesn't have any side effect. And actually, it has been approved by the FDA to be taken as a food supplement. Those who are above 40.
safe. Do epileptic patients who have tried it.
Nobody has tried in the epileptic patients. There is no clinical trial, but it is already done for the neurodegenerative disease and already approved by the FDA that the people above 40 actually they can take urolithin as a food supplement. Yeah. We did one is the external protein for the mitochondria. So I think the whole we think is that the mitochondria is very important, and that is one of the target. That is where the immune inhibitor it works. So the what we have done is that we have looked at the almost like 25 epilepsy model in Drosophila, and it cures all of them.
Great, sir. We can continue the discussion outside the speed on the itself. I told them to come on time. We can't take late. I'm outside. Yeah, you'll be outside. He'll be outside. Questions here. You go off. Yeah. Happy. Over tea. Come. And we are going to get back here at noon. Yes, go ahead. Come. Yeah.
That was the first but you will have we have just one, and we are working on multiple all these. One thing we have to we have that is I will solve is your Just by looking so, and then yeah, we should get it knowledge. Knowledge. No. It's all from it. That was what it's not easy. My students 5 years ago. Stabilized. Then the synapses, teaching. Synapses are very, very fast. Especially once the neurons, the motor they develop now, then you see, then development the motor it's going like 2-3 months. They are not going to that. It doesn't. Yeah, so right, right. Now the neurons modify itself because of and so the neurons, the protein will remain for 3-4 months because it takes up to say 1 year. So for 1 year, you have taken course in 3 months, like 1 year. That's right. Yeah, it's very difficult. So, and what we have shown is I think so. What we have to get cell that we have been able to look at autophagy, we have to look at how aggregations can be removed and exposed. So
Can it be also related by lifestyle changes?
you can. Well, I'm trying to do like yoga. You see, some things we see not in methodology requiring exercise. Right? So I'm saying this is because you see at the end we always want to be contributing something to the society. We are because our bodies requires excellence.
Also, without any more difference.
No, but remember all the things happen in medicine. Actually, should not be even
Similar to you, working on your own.
no, yes. You know, for example, that people look at the blood thing and they identify alarm receptor, the receptor, and the target of the in our case, electric model, alarm receptor didn't
Try to introduce or instill the government, like introduce that to make sure that
yeah.
ಆಗ್ತಾ ಇದೆಯಾ? ಹಾ?
Can I actually just download on the computer if net is working? I have it on my slides.
You can download to that.
Have you had some tea or something?
I'm just trying to.
Yeah, yeah, something.
Yeah, also going now. Thank you so much.
Next.
Yes. I just logged in here. It was. Yeah, so I have the presentation on it and I can present now. This is what we do. Okay.
So you share this.
That's okay.
Here. Screen. I use.
So let's end the audio start out and just.
Yes, yes.
I'm done with it. Yes.
This.
Just waiting for my object. Yes. Let me just Okay. Sure.
Okay.
No, no.
Okay, if nobody's outside, right? Although it's 3 minutes more. So what I'm going to do now is before I invite our next speaker, just to tell you what the next two talks are going to be. One is by our speaker Sethi from the IIIT in Delhi, and I will leave him to introduce himself. Right? And then after half an hour, we have a talk by somebody who's actually going to wake up and in Canada, in Newfoundland. Yeah, and it's 8 hours behind, so you can and you know what time it is for him. And he will speak about his work on peak genomics, and there's a parallel to that talk and how we look at human migrations. So, and then we'll have lunch. I think the things like certificates and so on that have to be signed will be done later, right? Around lunch and post lunch. Okay, so I invite Dr. Desh and please introduce yourself.
Thank you, Mr. Kastikar. I'm delighted to. Thank you. I'm delighted to be here. And I just realized that this is an audience which I think is very young and also coming from the background of biology. So I come from IIIT Delhi, it's an engineering institution. We are hardcore computer science institution, so you will see some technical details there. But my personal background is in medicine. I did my BBS and then did a PhD in population biology. For the last 15 years or so, I've been working in the space of data science, machine learning, and now AI in healthcare. So I should be able to connect with you, even if at some point it gets technical. I think I should be able to bring back the biology and the sense of it. Okay. So very glad to be speaking with this audience. I'm going to be speaking on genomics in the age of ChatGPT. Everybody has heard about ChatGPT. What is ChatGPT? These are types of large language models. Language models that have billions of parameters to train with, right? So, for example, y is equal to mx plus c is an example of a linear model with 2 parameters. Think about billions of parameters connected to them in a complex manner, and that's what these large language models more detail. What they could teach us about COVID-19 strain evolution. I'll be speaking mostly about work from our lab investigators. I'm an associate professor in the Department of Computational Biology at IIIT Delhi, and also the founding member of Center of Excellence. My lab's research largely focuses on AI and large language models as a part. Predictive modeling for ICUs. I'll not be talking about this today because this has very little genetics or genomics to it. Policy models for public health. But what I'm going to be talking about is knowledge discovery and application of genome statistics. The common thread behind all of these are use of AI and learning. So we address the data gap, the knowledge gap, policy gap. I will not really stress a lot on it, but my focus is on the lack of data and how can we use non-conventional sources of data to aid healthcare decisions. Also, once we have data, we need to convert that data to knowledge. Once we have knowledge, how do we really build decisions? All knowledge is not actionable. So knowledge that can be used to take some action. For example, what should we do about COVID-19 space? So I'll skip this because this was mostly background to my work, but I work with aims in the ICU space to build models that work with large data sets such as 2 million patient hours of continuous monitoring data collected at AIMS. That includes text data to make predictions about each baby, each child. Are they going to be crashing in the next 24 hours with sepsis or shock and so on? Right? So we make use of AI to use this window of opportunity to make predictions about patient outcomes. Okay. These data sets include imaging data, text data, and so on. And these have been validated in more than 200 ICUs across the global settings. And essentially, we were able to show more than 90% accuracy in many of these ICUs. So it's more than 200,000 patients across the globe, 200 ICUs, and our models can predict whether this person is going to go into sepsis or shock up to 24 hours. Why am I giving this background? Because the same kind of modeling setups will be used going forward when we start looking at data and start making predictions about the future. And that will be about COVID-19 scale emergence. Okay? And we'll be using models that are similar in their approach, which make use of language models to read the genome. These models have been deployed at AIMS and so on. So let's skip to the next one, which is the main core of our discussion. So language models start with something called a vector space. Some of you may remember from your high school math that vectors have quantity and a direction. Right? So basically they have magnitude and a direction. Vectors can be added and subtracted. How can language be treated mathematically? How can you treat words mathematically? Is by embedding them in a vector space. What do I mean by that? You can represent words as vectors. Right? So for example, if the word king is here and queen is here, you can actually calculate the relatedness between the king and queen vector by looking at the cosine similarity, which is the cos theta, right? Because if you remember, if the vectors are orthogonal, they have no similarity. If the cosine is 0, if the theta is 0, cosine is 1, right? So they're absolutely similar. So it can these representations can start giving these relatedness between words, right? So and then you can also, for example, add and subtract vectors. So you can actually do algebra like king minus man plus woman is equivalent to taking the king vector, subtracting the man vector from it, adding the woman vector to it, and that resultant vector will lie very close to queen. Okay, so basically that's a famous example of the paper called Word to Vec, and that was a paper essentially in 2014, which provided this kind of intuition behind vector space models, where we say that represent words or tokens. I'm talking about words, but we can even go to tokens such as tokens, where you can have sub-word level vectors. Okay, represent vectors or tokens, words or tokens as vectors, and words with similar meanings are related. They can be related through the mathematical operation of cosine similarity, and the weights are shared, right? So king and queen has shared weight because both are royalty. Okay? So if I have a billion words, I can create a vector space which is not a billion dimensional vector space. It may be only, let's say, 500 dimensional, even 36 dimensional. Why? Because we don't need 1 billion independent entities. We know that king and queen are related. So there's some sharing of information. This is called compressed representation. Okay, so compressed representations can be learned from data, if you have billions of words, millions of words and documents, these can be learned by training neural network models, which are AI models, which allow you to get the context. The word-to-vec paper basically, I'm not going into a lot of detail, but essentially works like this. You take the middle word, right, and you use a context to predict the middle. Okay, think about an AI network, a neural network, and you have, let's say, 10 billion words in Google News corpus. What it will do is, it will go through each word. It will look at the context of each word. So here, the word is, central word is jump. It is looking at the context of the word, right? And these models are predicting what is the middle word given the context. Okay, this is called continuous map of words, variant of the word. And this, since it happens over billions of instances, the model actually gets very good at predicting. So let's give this context and target we already discussed. This is how the neural network architecture looks like. Again, what you're trying to do, you're looking at the context surrounding words before and after, and trying to predict the correct word. Right? So you have a neural network which looks at the surrounding words and predicts the target. Okay. The other side of thing is you can also look at the middle word and predict the context word. Right? That how can I look at the word jump and what should come before and after jump? What are we trying to do? We are trying to think about generative models. We are trying to create language using a model. Just like ChatGPT creates a chat for you. How does it do that? It looks at some word and it says, oh, this should be the next word. So this should be the next word. How does it learn that? By doing something similar, not exactly this, I'll come to what it does, but something similar by looking at the context. That in these millions of documents, billions of words, right? What was coming before and after this word? What should I give the highest probability? That's what is used here to basically learn a model that predicts the context, right? And this also performs pretty well. This is again from the Google paper. I'm not going to explain a lot, but what you do is now basically you are taking the middle word, so it's like a verse. Then you actually predict the context. Okay. Now this kind of architecture we used for the first time to understand the COVID-19 variants. Why? Because we had a lot of data available to us in a portal called GISAID, where COVID-19 sequencing was done by many countries. India was also doing it. There's an so-called consortium for every patient. We isolate the DNA, we sequence the virus, we deposit it in the portal, not for every patient, but I'm making it simplified for you. But for many of these sequences are deposited on a portal, national portal called INSACOG, which then basically makes it GISAID, which is a global portal. Long story short, across geographies, more than 3 million COVID-19 genome sequences were available to us. Okay, so we said, can we train language models with sequences, right? Because sequences are also consisting of codons. Codons can be thought of as words. What are we trying to do is we're trying to learn the grammar of a sequence using the language models. Just as the way I basically showed people, right? So for example, skip-learn approach, we look at this particular location, we look at the context, and we try to predict what the context is. Basically, all the codons in these 3 million sequences. Okay. So we took COVID-19 genomic sequences from GISAID, trained word-to-vec language models. Okay, and then we did some quantitative analysis. These were from, sorry for the resolution, 17 different countries, of course, including India. And then we did some qualitative analysis that I talked about. This is how we only look at the spike region, because training a model in the spike region, which basically is the region which the virus uses to bind to the receptors and get in. So you can look at, this is the length of the spike region of the COVID-19 virus, and essentially you can see that where are all the mutations happening. So this is just qualitative. Where are all the mutations happening? Nothing new, but just to give us an insight on what are the mutations. We've not discovered this for the first time. But what we did was we used the models that we learned in the word-to-vec space to understand that what are those grammatical features that can help us predict what will happen next. Okay, because these mutations are known not to be happening independent. There's some grammar of these mutations. If I'm a virus, I might want to, let's say, optimize for some function. Basically, I might, for example, the mutations accumulate coaxially along the. So that hypothesis that, and I'll show that how this actually hypothesis turned out to be correct. And we were able to take out features like entropy of certain features using more grammatical dimensions, which were leading 2 months ahead of time before the spikes came. More on that later, but just keep that. Okay. So these are the data training set. When you train an artificial intelligence model, you don't train with all the data. You put some data aside for testing. Okay. You take some training data, you build model, and then you validate on the data that is outside. So our training data was about 3 lakhs, right? And the prediction was 2.5 million, total 3 million sequences. Okay. And you can see the countries represented. This is the world, India, and Africa. What are these plots? These are called T-snake plots. Okay, so to an expert audience, these are quite obvious. But what I'm trying to do here is, I want to characterize the sequences that I can visualize in 2 dimensions. Okay, I'm working with millions of sequences, millions of words. How do I represent these words in 2 dimensions? So these are called T-snake embeddings, which basically similarity and the dissimilarity of shapes is what we look for. What do these tell us? These told us that how countries were similar, for example, India or UK changed over time, how USA changed over time, and so on, right? And over different months as well. What are we doing? We are trying to understand that does AI even learn something meaningful on this data, right? It may just learn something which is garbage. Right? So we just trying to first see, does AI learn any patterns that should exist? So this is that line. So for example, you can see that did capture patterns. Australia, for example, forms a cluster, England forms a cluster, China forms a cluster, and these are in different spaces. But also they will be captured how the temporal evolution is. Which means that our grammar, AI model was actually learning grammar as it changed across. This is not done using a blast analysis. This is done using cosine similarities that I showed you earlier. Each point here is a string. Okay, isolate. Essentially, the isolate has a vector, right? And another isolate has the other vector. How similar are these? If they're similar, they are in similar space. If they're not similar, they may be different apart, separate apart. Okay, so this is what is happening. Listen, we do see that France and Germany, for example, come together in this vector space representation. USA and Canada, which are geographically close together, they also come together. Again, qualitative, right? Nothing quantitative. Gives us confidence that the grammar has some spatio-temporal. Then we go to statistical analysis. We ask the question that now that we have learned grammar, what it has allowed us to do is go from billions of words to a lower dimensional compressed representation with shared weights that I talked about. Right? So let's call it embedding, which means that now I don't need to be looking at millions of dimensions or millions of photons, but I'm only looking at 36 factors, 36 dimensions. That's the level of compression that you were able to find out. That's the level of compression you can achieve it word-to-word. 36 independent dimensions on which we characterize each isolate. And then we asked that any of these dimensions, is there any dimension which changes ahead of searches in cases? Because we had that data from Johns Hopkins University and we had this screen data, we could ask the question, as the dimension changes, which is shown here in blue, it's called LS32 entropy. LS is latent space dimension 32, and the entropy means how fast it is changing. Okay? So the entropy goes up and starts falling. This happens 2 months ahead of time before the peaks come. This is true not only for one country, this is true for all these countries, statistically speaking, which are present. This analysis basically tells us that grammatical changes in the virus can be captured using language models, which are happening early. Okay, so it is an early warning system, pandemic surveillance system that can be put in place, which captures these case surges in certain different countries. Okay. So there's a hypothesis. Basically, the virus explores a set of mutations and then exploits, and then it starts exploiting, the entropic starts falling, and that's when cases start rising. But that's a hypothesis. We are talking about the empirical observations. We validated this using machine learning models. That was statistics. This is machine learning. We made use of random forest based machine learning models. Again, entropy of dimension 32 comes out on the top. Okay, the more high you are here, the more important that dimension. So there's 36 dimensions that are represented here. 36 grammar dimensions of the virus, and one of these 32 dimensions is actually on the top. Of course, this one is country. So most predictive factor was country. When was, if you're in India, we can predict when is the case of this happening because we were following a different trajectory than USA. Right? So we see that these dimensions actually work. Okay. And then we did some biological characterization of these dimensions because what they do not allow us to do is that these dimensions are compressed, so they don't tell us where exactly it's changing. We can just say that this particular dimension 32, for example, has a high weight from this 3 multi AA. Okay, we don't know where the A, where in this confusion, but it has a high weight from, which means that we can interpret going back that okay, is TAA known to be something which is important, for example, for the variants of concern, delta variant. So we did that and we find that actually was the case. Right? So this is in this case, for example, dimension 32 CPG was present in the data. So and then this is listed in the paper. Gives us more confidence that what the models are seeing as biological. Okay. And then basically we created a dashboard out of this to give to public. We circulated, and this was actually published as a paper as well as the Vigyan Prasad, which is a national magazine report also. I also wrote a piece about it. Where basically the dashboard showed how are the countries doing in terms of case surges. And we were in most of the countries 2 months ahead of time able to predict the case surge. How well it was used by governments, but then it gives the interpretation. So the dashboard is called strain flows. Model is called strain flow. All right. So now comes the. I know it's only 12 minutes remaining, but I think if you understood this, the next part should be easier. The next part is on ChatGPT. ChatGPT makes use of the next level of model that I just presented to you. The level of model that I presented was called word to word model, you predict the context and so on, but we're still going word, right? This is not how babies learn language. When babies learn language or kids learn language, basically this paper by Maswani and Google Brain, 2014 paper, attention is all you need, was the famous paper that is at the back end, all that you are seeing today in chat. What is attention? Attention is, I don't go word by word, I pay attention to the entire sentence. Okay, and then as kids or as when we learn something new, we look at chunks of information, and then we basically derive meaning. I'll not go to the back of it, but what exactly it does is, if I have to predict the word she, right, I would look at the whole sentence. How is your sister? That's how it should not be. Thank you. He is fine. Right? It should learn that sister is she. Okay. So basically, this context is being learned by something called attention mechanism, where every word being predicted pays attention, receives attention from the sentence, not a word. Okay. I'll skip the math of it, but the context of the sentence context drives the. What it allows you to do is something very interesting. Look at the sentence. The animal didn't cross the street because it was too tired. Okay. Now, if I trained a transformer model with it and asked, if I look at the sentence, think about yourself as a baby, it was too tired. Then where is the maximum attention coming to it from? It was animal. Why? Because the animal was tired. The animal didn't cross the street because it was too tired. It refers to animal. I changed one word in the sentence. The animal didn't cross the street because it was too wide. Now the attention it is coming more from street. Street is wide. This is the backend of transformers which drives GPT. Now basically one word change leads to a completely different prediction or less. This is called the attention mechanism. And we extended it. I'll skip all the math of it, how it works. But what we said is, can we do something similar for the genomics systems? Again, COVID-19, because we knew the limitation of our previous model. We can't say where is the AAC coming from that is contributing to the right next. We can't be predictive. That's what we basically did. We calculated, we trained the transformer models which are at the back end of chapter kind of. And then basically we, this is unpublished work yet, it's under submission. So, but I'm very excited to present the findings that we see that there are hubs of attention. What does it tell us? That in the genomic sequences, there are certain locations which are receiving more attention. So if I could do some surgery, that's in design, something like that. Surgery means, surgery. These are the places which could tell us something. Okay. So basically, we developed the strain forward model. We transformer and strain flow, basically strain forward. Why also another meaning for strain forward that actually can allow us that if I change this and change this and change this, what could happen? So we are doing that on demand formation of insights. So this is what the workflow is. I'll skip some of this, but what we find is that again, this is the length of the spike region. We see that attention is not equally distributed. There are some regions, some parts where attention is low, some parts where attention is high. So there are hubs of attention. So if you think about these as networks, right? Each codon being connected to the codon to tell if I change this, what would happen to that? There are hubs of attention within these things. Okay. And these hubs of attention is what we basically can explore. We show some similar insights from countries, right? That we see even fine-grained information that we were not resolving earlier in the strain flow model. Now we're able to actually resolve even further. Why? Because now we have even a stronger 768 dimension space. I'll not bore you with the details. Long story short, we can predict that if I have a certain codon here, right, and I don't know what that codon is. I can actually make use of the context of the virus to basically predict what might be that codon. That's called mask language. Okay, and we achieve more than 90% accuracy. This mask language. We believe that this is an important advance to basically understand how virus evolution also happened and also how vaccines could be designed because it allows us in silico scissors to actually work with this. There are some emergent properties which the model never saw. Synonymous, non-synonymous mutations. We characterize the model that if some mutations have happened, you may argue that the model is seeing how frequently some position is changing and predicting, right? But the model never knows that if there is a certain codon, what amino acid does it translate, right? That's biological knowledge. It was able to capture the biological knowledge, the relationship between between synonymous and non-synonymous usage and hubs was not uniform, was not random, it was non-random. Hubs are protected against non-synonymous mutations. Let me just summarize by saying that which this plot captures. These are early findings. So we also validated this using thermodynamics calculations, where we basically show that hubs of attention are protected against. So the model is definitely learning some biology. It's not just learning some statistics of sequences that we are giving. It's actually capturing the mind of the virus, so to speak. We are still early stages with this, but I think that's something which we are very excited to be talking about. And that's what I was trying to show, that how stabilizing and destabilizing mutations and hubs are different. So they are actually okay. So point mutations will do harmless but more stable in nature. So what we are doing now, this paper has been submitted, and we hope to see that this will be received with some enthusiasm. But what we are doing also at this point of time is strains are just one dimension of pandemic. There's also mobility, lockdown, also socio-demographic factors that govern how pandemic should be handled. How other features such as vaccination, etc. So we are actually building an integrated model using these transformer kind of approaches, looking at context, integrate all them into a pandemic preparedness platform. So that is what we are trying. Based on which integrates what I just showed to you earlier, strains information, mobility information, background health and demographic features to give insights. This is just illustrative, like what we saw in strain flow much earlier. So we have a portal where we host these insights. It's called Federated Health Platform. It has these models that are hosted and are openly available, or at least for India platform, openly available. And you can also explore other models like evidence flow maxes that we have created for other kinds of data. And we also have a coding data student with more than 6,000 data sets have been curated using AI approaches so that we can curate more insights. If you're a student excited in working with science, trying to look for data sets, then that was a that may be useful. So I'll stop there. Those are my others, students who have been working on the collaborators. But
genomic sequencing is a very expensive process. So how potential this ChatGPT can be in making the expenses?
Very good question. I think I never thought about that. But for example, if you actually know which areas to target, what are the important links, then you don't need to go through. That's one way I did not think about before you ask this question, that these kind of models can help develop better strategies for genomics, which can be made less expensive. Okay, so that is one way, because we can actually focus on the right parts. The other side is that genomic sequencing is expensive because the costs are coming down. But this was just an illustrative example that how AI and genomic sequencing should come together to actually reduce the cost, because the demand goes up. Right? So for a lot of things that we are now thinking about, for example, wastewater surveillance, right? Because COVID-19 is just one pandemic. How many genomic sequences have been deposited for MERS? Hundreds, not millions. Right? For Ebola, maybe somewhere in hundreds of thousands. TB, maybe thousands. So we need more data, genomic data, right, to be able to really develop strategies for pandemics. So I think that's the other side of the things, that if we actually create such examples, cost of genomic sequencing can also come down if there is more and more demand.
One more question. Is there any risk?
I think maybe one question, maybe I can.
Yeah,
we can
I was just curious, when you use these kind of AI-driven models, let's say for applications like genome assembly, phylogenetics, these are.
Phylogenetics is known. Genome assembly, I have to explore. I am sure there will be but I think that will be a
but
good.
the same, right?
I think I was also thinking about what Dr. Dr. was saying in the morning, because sequences that we have actually going beyond pathogens, looking at what are very important. So I think all those are applications which are potential. And
Very important to say AI is not biology. So have you tried, see, there can be several groups to try and get a particular strain. Have you tried taking the initial strains, skimming the middle strains, and then using the previous strain, then make sure that same
we see that at a macro level, and not at the micro, they're not explored at the micro level. We look at how does the hubness change across. So basically, what were the features of the hubness of attention across time as trains. We do see it there, right? So and those were done independently. So yeah, we can remove the initial memory. It's still see it there. But we've not done that at a micro level where I see a codon level what happened. That's an interesting question. Actually, we have been trying to address that, but I couldn't find students who would buy that argument that can we really train models which learn to play chess to really evolve a virus using these things. Yes, so that's reinforcement learning models to actually learn in what happened.
Okay, last question, then we.
So when so many inventions and inventions that are happening randomly in the world, and which these models predict the search in different countries. How precise can they be without using the real-time data of first data?
Great questions. These were not, I mean, these were mostly, we did prospective validation, but these are mostly retrospective data. Right? So we are actually looking at retrospective data, what data were deposited in that year. We didn't have real time data. None of the countries in the world have real time data. So these are mostly retrospective, and then we are making predictions about what happens 2 months time. And we were prospectively validating, yes, this happened. Right? So we do see that it's not completely random. That's where we show those top weighted stuff. But I can talk more about that.
Yeah, so we can, here at lunch, here beyond lunch, so, you can interact with us.
Thank you.
Thank you so much, Bob. So, now we, jump overseas to our next speaker who is joining us from Canada. So, are you there, Steve? Oh, there you go, yeah, I was looking at the wrong panel. Good to see you. I can't hear you, you have to unmute. Unmute.
Yes. Can you hear me?
Oh, yeah, it needs volume, we need the, increase the volume of it.
I'll just talk for a little bit until you've got the volume.
So Steve,
Okay, there.
while we are fixing the volume for you, I'll tell you what we are doing here. Each speaker gets half an hour, and in that time you can also briefly introduce yourself. And at the end of the half hour, we will take a few questions. So to make it easier for you, stand at the mic and harvest the questions from the audience and repeat them for you. And you can answer it. Right? So now you can start speaking and we'll see the volume is fine. And the volume
Okay.
is over. Okay, great.
And let's see if I get this properly. Thank you everyone for the invitation to speak today. It's 4:30 my time in the morning. And okay. So can you all see the PowerPoint?
Yes, we can.
Okay, great. So let's do this. What I wanted to talk about is some work that was inspired by my being an editor on the Journal of Genetics, and as I will address as I get into this, that I had occasion to review a paper for the journal that was on honeybees and on a particular marker that was being used to distinguish between subspecies of bees. And I will introduce the talk by saying that I know absolutely nothing about honeybees, but that does not stop me as an editor from having an opinion about honeybees. So a good American expression is, I don't know anything about this, but I will tell you what I think. The question that arises is actually very simple, is when you have a particular bee, you seed a bee, and then you want to know, is the bee a European bee or is it something else? The accepted theory on honeybees, and I'll remind you what the bees look like, is we have on the left in a cage a marked queen. And so she's in a cage, so she will not be able to escape from the cage, and she's also tagged, so we know which particular queen she is. And then the bees that we usually run into is the one all the way on the right. That's the worker, who is a haplodiploid female. And then in the middle, the one that we very seldom see is the male drone. So the species is haplodiploid. There are a number of species of honeybees, and the question arises, how are they related? There are, within the genus Apis, which is the common honeybee, there are three subgenera which are known as the giant honeybees. And the giant honeybee will be familiar to you in India because the species Apis cerana is a domesticated species in India. It's one of the two domesticated species for honey and wax and other products. That's indicated by the blue range. We also have the dwarf honeybees, and the dwarf honeybees extend from the greater south across into Africa and across the Arabian Peninsula. And lastly, we have the cavity nesting honeybees, and the cavity nesting honeybees are again found mostly in Asia. And the one exception to this is the Western honeybee, which is found in throughout Europe, into the Middle East, and down into most of Africa. The question that arises in the evolution of bees is, first, what are the relationships of the three subgenera, and more particularly, the question of what are the relationships amongst the 30 or so subspecies of the Western honeybee. Now, this is important because there's a great deal of variation amongst the Western honeybees for such things as what is the quality of honey, what's the flavor of the honey, how aggressive are the bees, a number of things that are important in in apiculture, the raising of bees. For example, one of the things that has happened by accident is the familiar story of the Africanized or killer honeybees, where an African form, which I will talk about in a little bit, African form Apis scutella, some drones were imported to South America because the quality of the honey and the quality of the wax was thought to be better. Those were imported into South America, crossed with local bees there. And unfortunately, this produced a very aggressive form of bee, as well as making better honey. But those bees have escaped, and they're called killer bees because they can actually kill people, especially younger children, when they escape. And the Africanized bees have been spreading northward. So there is some practical interest in knowing what the relationships are. The received view for a long time has been what has been called the out of Africa hypothesis, which is that the bees, Apis mellifera, originated in Africa and then spread northward through the Middle East, through southeastern Europe, and into northern Europe. And in northern Europe is where the major center of cultivation happened, and most of the international stock is taken from Apis mellifera in Europe. The questions about where those bees came from would seem to be suggested by this picture, that if we look at the dwarf honeybees are the only ones that extend as far west as Africa. So the historical assumption has been that the Apis mellifera is related to the dwarf honeybees. And that's what we wanted to find out. So if I go to our method is that we're using bDNA. There's a little joke here. I hope it's early in the morning for me to be making jokes, but this is the bDNA form that we're looking at. And the particular kind of DNA that I'm looking at is mitochondrial DNA, which in animals is famously a small circular molecule and has been very widely used since the earliest days of DNA to look at evolutionary questions and phylogenetic questions. What I did was a meta-analysis of existing data, and the meta-analysis, of course, is that I simply went to GenBank and I looked for all records of complete mitochondrial DNA genomes using a couple of different ways to check for them. I started this work about a year ago and continued looking for sequences up through October of 2022 when I finalized the data set. This data has come out literally today in scientific reports. I can send a link to the board so that you're able to get this. But I did literally just get the final publication today. The region of the mitochondrial DNA that I looked at is just the coding regions. There are standardly 13 coding regions in most animal mitochondrial DNAs. Some of them are on the heavy strand, some of them are on the light strand. And when you take out the ribosomal DNA, the transferred DNA, and the D loop, which is responsible for replication, we take those out because those are often very difficult to align when you're looking at things that are moderately not closely related. That leaves you with 11,006 base pairs in the molecule, and those can usually be lined up pretty accurately because they are coding for the same molecule, and you have the codon frame to rely on. The outgroup that I chose to look at was a bumblebee, Bombus ignitus, and it just happened to be one that I knew something about, so I chose that one. The bumblebees are in a separate tribe, the Bombini, as opposed to the Apini for the Apis species. Of the Apis species, I was able to look at 9 species in the group. There are about 15 or so species. Not all of them have been examined for their mitochondrial DNA sequence. And then the one that is of particular interest then is Apis mellifera, which is a Linnaean species, the Western honeybee, or the German honeybee, or the dark honeybee. There are in over 30 subspecies known, and we have complete mitochondrial DNA sequences for 22 of these subspecies. Now, what I did that is different from what other people did is rather than looking at a single mitochondrial DNA sequence per subspecies, I took all of the individuals that were available for all of the subspecies there. And this amounts to about 78 individuals that are in the data set. There are multiple replicate sequences then from 8 of the subspecies. And my original interest in this is, well, I just wanted to see what the range of variation within the subspecies was. And that's where the initial problems started, is that when I said, I want to look at the type subspecies, Apis mellifera mellifera, and started looking at the sequences, I found that when I looked at the multiple replicates that were in the GenBank database, those animals were scattered all over. Some of them were very closely related. Some of them were very distantly related. When I started comparing them with other subspecies, there were all kinds of problems with the evolutionary picture that came out of that simply, and it would imply that Apis mellifera was highly paraphyletic or polyphyletic, that it had multiple origins. And based on that, then I also did want to look at more individuals. So this is an issue of what we call alpha taxonomy, and it's the most basic form of taxonomy, is that you want to get the names right. And the problem here is one I'll talk about later, and it's one that I have run into in being an editor for the journal, which is that if you go to GenBank, the names in GenBank are not always accurate. That in many cases, for example, someone will submit a DNA sequence, and they will just pick out the common name, the commonest name for that genus. And this is especially true of people who are not using the database for taxonomy. They're using it for disease organisms. They're using it for bacterial strains. They're using it for any number of things like that. They may not be worried about the taxonomy, and so they pick out any name that is available, and often they will pick out the type species name for this. So that was one of the first problems, was sorting out the alpha taxonomy, and that is something that emerges only by doing the analysis. The analysis is of three kinds. The first one was a maximum parsimony analysis. This is the one that I happen to like, because this is the way that I did my PhD thesis on a very large piece of paper quite a number of years ago. But this is attempting to explain the evolution of each base pair across the 78 individuals. Unfortunately, now we don't have to use paper. We can do that on a computer. So the maximum parsimony method will attempt to put together a evolutionary tree that has the minimum number of changes. And that's our criteria for choosing the maximum parsimony tree. A second method is the neighbor joining method, and the neighbor joining method uses genetic distances. So in this case, it is simply if you compare 11,006 base pairs in one subspecies with the same 11,006 base pairs for another subspecies, at how many positions do they differ? You express that as a percentage. You can also, with the modern method, simply express it as a count of changes. And there you are trying to minimize the overall distance that is required to explain the tree. And the last one is the maximum likelihood method. Maximum likelihood method is the most recent one to be added to the repertoire, because it is very computationally intensive, and especially for something like 78 individuals, it can take several days, as it did in my case, working on a laptop. It can take several days for the models to run. This is a Bayesian approach, and it is called model-based because you come up with a particular theory or a model of how A, C, G, and T mutates to A, C, G, and T, or changes to A, C, G, and T. And that gives you a 4x4 matrix. There are other things that you can put into that, but it is maximum likelihood is basically finding the least unlikely tree. And it is saying that for any particular tree, a whole series of events has to happen. And you compare trees by saying, well, which one, all of the trees are extremely unlikely, but which one is the least unlikely? So that's maximum likelihood. Our statistical confidence in this is a bootstrap method that you resample the 11,000 base pairs 3,000 times, and that is literally just taking the 11,006 positions, taking one position at random, taking a second one at random with replacement, taking a third one at random, and you do that random resamplings with replacement 3,000 times, so you come up with a new data set that's 11,006 base pairs long. And so the theory of that is that if your data are a true sample of an infinite data set that have the same properties as yours, how many times would you come up with the same set of relationships if you did a random sampling from that data set? And so that will be the bootstrap confidence. And the last thing that I did was to use a molecular clock calculation. And a molecular clock goes back to the very old days from my major professor, Alan Wilson, was the recognition that molecules, DNA molecules, protein molecules, often appear to change in a clock-like fashion, not a metronomic clock, but more of a Poisson clock or a radioactive decay clock. And the method that I used for estimating this clock was to take the maximum likelihood method again, add another constraint, which is that changes have to occur in a linear fashion so that it will not be metronomic, but the same number of changes need to occur over the same interval. You recalculate your tree based on that, and then that allows you to convert directly to times if you have a clock rate, which we happen to do. Now that's spending more time than I wanted to on this one. And so we'll just go quickly through the data. The first thing that I found out was the relationships between the three subgenera. And it turns out when you use a bumblebee as an outgroup, and mine was the first study to actually use a outgroup rather than just making an inference from historical assumptions. When we do that, we find that the dwarf honeybees are the basal group, that they are the most distant relationship or most distantly related group to the others. Then comes the giant honeybees, and then the most recently evolved group are the cavity nesting honeybees, and the cavity nesting honeybees include mellifera, Apis mellifera, our western honeybee that we're interested in, and that is basal to the other cavity nesting honeybees. So the behavior that makes honeybees useful is this cavity nesting behavior. And if you can nest in a cavity, then you can be trained to nest in a hive. So that's the advantage there. Okay. And so just to give you an idea what these trees are going to look like, the number of changes that are inferred to occur on each branch are given above the branch. So there are 335 substitutions which define the cavity nesting honeybees. So these are all molecular identifying marks. And the confidence in that branch is 99%. And if you look at the rest of the tree, all of the branch points, except for two of them, are in excess of 95%. And the only one that is lower is down here, but that's still a very good inference. So our basic group here is 99%, with mellifera being at the base. Okay, and this is what we wanted to know, is where does mellifera fit in? When we apply a molecular clock constraint to this, we get the same picture, and this allows us to infer time differences. And I'm going to go over this quickly, and that we're getting the radiations within the dwarf, the large, and the or the giant and the cavity nesting bees is all at about 7 million years, which makes it a late Miocene radiation. The nice thing about that is that is more or less what the bee fossil record suggests. All of those come together at about 10 to 11 million years ago, and that is still late Miocene. And I understand that there are some people that are going to be upset with me for suggesting that it's that recent, and a lot of people would like to think that it's much further back. For our purposes, for my purposes, the important thing is that when we do the same analysis that we did before and we add just a sample of the sequences from Apis mellifera, we find out that Apis mellifera So the type subspecies for the whole species is the basal species, and that is a new result coming out of mitochondrial DNA that has not been found with the other data. And as you'll see when I show you the distributions of these things, that gives the lie to the out of Africa hypothesis. And that the radiation here is quite recent in the Pleistocene, about 800,000 years. Okay, then this is a schematic version of all of the subspecies, and I haven't included all of the 78 branches, but I'm indicating groups by species. And this is an expansion of what I just showed you, is that Apis mellifera is basal. This is the European bee. There's quite a lot of diversity. One set of lineages here, another lineage here. And the most recently described subspecies is a species from China and the Sichuan province. Only a single individual has been looked at there, but that animal is quite close to the European mellifera. And there is some question as to whether that Asian distribution is ancestral or whether it is derived, and my analysis would suggest that it's derived. We also have a total of 6 clades that we can make out, and the 6 clades are very nicely arranged biogeographically. There's one that occurs in Southeast Europe, which is dominated by Ligustica. Ligustica is the other species that is used for honey. So it's the Italian honeybee, and some people prefer that. It's a nicer bee to work with. And then we also have an Asia Minor group. The Asia Minor group are all very closely related. And even though these things are called 3 different species, one of the things I want to emphasize in this presentation is that you're looking at pairwise subspecies differences of 1, 2, or 3 base pair substitutions. And if you look at another subspecies, Ligustica, well, the differences between all 3 of those subspecies are less than what you see amongst multiple representatives of the one subspecies. So again, this is an advantage that you would not see if you only looked at a single individual. We also have what I'm calling a Levantine nilotic Arabian group. And this is in the Levant, in the Middle East, goes into Saudi Arabia, goes into the Yemeni coast, and also occurs in Syria, also occurs in the Nile Valley of Africa. And so this is the first introduction of honeybees to Africa. Then we have a subspecies that is found only in Ethiopia. And then we have another subspecies which used to be only found on Madagascar and Malagasy. And one of the results of my study was to demonstrate that what somebody else had said was a Apis mellifera is in fact much more closely related to this Malagasy subspecies. It's found in Kenya, so that subspecies is no longer confined to the island. A very interesting group here is that there's a group of Mediterranean bees. They are found in Spain. They're also found in oases in North Africa, and they're also found on some of the Mediterranean islands, Sicily and Malta. And again, these are all very closely related to each other, and there's less difference between all 5 of these species than there is within Ligustica or that there is within Yemenetica. Okay, and then when we look at the South African bees, the sub-Saharan bees, there are 4 subspecies, excuse me, that are found there. And the 2 major ones are Scutellata. This is the most widespread of the African bees. There is also the capensis, which was originally described from the southern tip of Africa. And if you can follow the colors here, you see that capensis is basically distinct from Scutellata, except that there are a number of instances in which a bee that the literature says belongs to the one subspecies is actually more closely related to the other. So we have an exception here, a number of exceptions there. And in one other case, there is a bee that is found in the mountains of Eastern Africa. And this group, two of them are assuredly Monticola. And then there is a third one assigned to Capensis, which also fits into that group. So again, we have the difficulties of the taxonomy. There's some suggestion here that there is confusion about which particular species these go into. And the radiation of this group is quite recent. It's 200,000 years. And then if I put all of this together into a phylogeographic model, then we get what I have called the scramble for Africa. And things fall apart, to use another Africa novel, things fall apart with the out of Africa theory, which is clearly from the mitochondrial DNA. The subspecies has originated in Northern Europe. It has then spread to Southeastern Europe, and these are where all of the specimens I've got come from. It spreads from Southeastern Europe into Asia Minor and further east, also up into Kazakhstan, also out into Persia. And then it spreads by way of the Levant, down the Red Sea, across into Arabia. This is the Yemenetica, which is quite widespread, and it's found not only in Saudi Arabia, but it is found in Africa. And then there are species that are found only in the Nile Valley. And so we see things extending into Eastern Africa. And then our Ethiopian bee is the next southern population, the Ethiopian bee. Our unicolor, which was found only on Malagasy, and now we find one individual that fits into the unicolor picture there. And then our Scutellata capensis complex is found throughout sub-Saharan Africa, and various things are called Scutellata or capensis in various areas, that being one of the outstanding areas to look at. And then our return to the Mediterranean and Europe in the last 50,000 years, and most of these species being only a few tens of thousands of years apart. So again, this reverses the accepted theory that bees evolved from south to north, and there is a very clear biogeographic trend to go from north to south in an absolutely linear order that makes far more sense than the other theory does. Again, putting times on all of this, won't go through this. If you're interested in bees, this will be of interest. If you're not interested in bees, you won't be. We have the diversification. Once we look inside Mellifera, the diversification of bees is about 720,000 years. The Eurasian group is about 134,000. And this is roughly, and if you want to say that, just take the first two decimals for accuracy, the Eurasian radiation is about 130,000 years ago. The entry into Africa is about diversification from the existing Middle Eastern lineage is only about 160,000 years. So this is a separate invasion of the Nile Valley. And then when we look at the entrance into Africa, that's actually about 550,000 years ago. The first group to be established is the Ethiopian, then the Malagasy. Then when we look at the sub-Saharan radiation, this is about 250,000 years ago. And the Mediterranean radiation is only about 100,000. And again, if we look at those five species, they're more closely related to each other than is this group up there. Okay. Okay, so to quote that well-known authority on bees, Winnie the Pooh, I have been thinking and I have come to a very important decision, is that these are the wrong kinds of bees. And in my opinion, and it's supported by this analysis of the data, one of the difficulties that has happened in previous studies of the bees is that they have used the wrong kinds of bees. And these are the two most recent meta-analyses of mitochondrial DNA. And this is by Boardman, Imenifar, and et al. And that group I want to acknowledge as having done all the primary DNA sequencing work. So I could not have done this work without using their data, but I just want to extend the interpretation that they made and point out what some of the difficulties there may be. And then Eric Tehelka and co-workers also did a meta-analysis, and they are particularly, as I'm going to just show briefly, they're particularly plagued by the particular individuals that they showed. The first thing, so the ones that are in the Tehelka study are T, the ones that are in the Boardman and Imanifar study are in B, and that's the total number that they looked at. So it's about 18 in either case. The first problem that comes up is that the sequences that they use to represent Intermesa and Syriaca, where Intermesa is a Mediterranean island bee, excuse me, that is a North African oasis bee, and Syriaca, there's something wrong with the data. These are what we call long branch attractors. They're far more similar to each other than they should be, given that one is found in Syria and one is found in North Africa, and that they have a particular region late in the sequence in which they are literally, they appear to be identical over a long run of the DNA sequence. And so what I'm going to suggest is that these are defective sequences and we should not rely on them for anything. And unfortunately, both Tehelka and the Boardman group have relied on them. The other example of Syriaca in the literature is here within the Yamanetica group, the Arabian bees, and you see that it's very different from this one. And the other example of Intermesa, the North African bee, is over here with the other Mediterranean bees and very distantly related here. So one of the problems is using the wrong sort of bees. Second problem that shows up is in Boardman is what did you use to represent the sub-Saharan bees, Scutellata and Capensis? Well, there are a lot of different bees to choose from. Boardman chose these two, and unfortunately, these are bad examples of their subspecies. Okay, now that is going to be more obvious when we look at the Tehelka species, and here, by the perfectly horrible luck, the example of Scutellata that they have chosen is one of those misnamed Capensis bees. Their true Capensis bee is over here, which is part of the Capensis group, but these two then come out in their analysis as being far closer to each other than they actually are when we look at the total range of variation within the group. Okay, so this simply shows that if you don't get your names right when you are choosing your specimens, or if you choose an atypical sequence to represent the subspecies, then you're going to get a wrong answer. Comparing these with nuclear DNA studies that have been done, again, there is a sharp contrast between Carr's Things Fall Apart, the Scramble for Africa theory, and the previous studies of nuclear DNA. The classical classification of bees is there's a mellifera group, an African group, an Oriental or Asian group, and a continental group. And this is called ACMO, or in fact, when you look at the DNA arrangements, the backbone of the phylogenetic tree is MAOC broadly. And then the out of Africa or out of Asia or out of North Africa theories. Which of those you choose depends on how you root the tree. So in the classic study of this, the tree is rooted within the African species. And one of the challenges to this, it's not rooted within the African group, but it's rooted between the African group and the Oriental group. And so this was used to suggest the out of Asia group. And then in the most recent study that's been put forward, they rooted their tree within the African group, but they rooted their tree so that the Asian portion of the of that African group, the ones that are in Arabia, that's the outgroup. And so that gives you an out of Arabia hypothesis. And the fundamental problem is first that this is a different species order than I get. The way the root has been placed is by using a very distant subspecies Serena. And we're just about out of time. So my conclusions, and these are my conclusions for Journal of Genetics editors, is as reviewers, we're all, I don't know anything about this, but I'll tell you what I think. So we're presented with papers on taxa that we don't know much about, but we have to develop some interest in that. You should always get a second opinion when you're looking at the names of things. Don't believe everything you read just because something is identified as a particular subspecies. Check it against the other data, and that names matter, and that alpha taxonomy remains important in this century, and GenBank is not infallible. And the molecules don't lie, but the taxonomy curation is sometimes flawed. And I apologize for going over. Yes, okay. So there we are.
Bringing the buzz into the mini symposium, I think we can take one or two pressing questions. I'll stand here and sort of transmit the question to you in case you can't hear the speaker who asked the question. So anybody have any questions on speed taxonomy and its revision? Yeah.
When you say something else, things populations.
Okay. Could you repeat the question? I didn't hear the question.
The question is, are your conclusions supported by non-mitochondrial DNA-based studies? Could be, the nuclear DNA studies or other things, non-biological studies also. So how does that bear on your conclusion?
Again, this is that the non-mitochondrial DNA data are we reach a quite dissimilar solution to the taxonomy problem by looking at those. In several cases, they simply do not make sense. In particular, there is a desire by the people who have done the classic work to preserve the model that there is everything that is in Africa has a common origin. And what my data, or the mitochondrial DNA data, there's somebody else's data, what those data clearly show is that the Mediterranean species or the North African species have a separate origin from the rest of the African species. What I'd like to suggest is that where this is a haplodiploid species that is spread by queens, so it is the movement of queens is the only thing that is going to allow the species to spread, that perhaps a maternally inherited mitochondrial DNA molecule is more reliable than the nuclear data. The other limitation of the nuclear data is that the nuclear data only look at 10 or 12 of the subspecies each. And so one is there is never a complete picture from the nuclear DNA data. And the other short answer to your question is yes, there is a difference. And that is something to be sorted out now that my analysis is before the community.
Okay, thank you for the answer. Yeah, one more question.
Is there any on anything with mitochondrial?
Okay, this is not related to the migration of bees, but this is inquiring about whether there is any genetic imprinting in mitochondrial DNA. Is there any clue? Yeah. I can answer on this behalf. I don't think so, but if you disagree with me, Steve, please speak up.
Okay, and please repeat the question.
Yeah, the question was, is there any evidence for imprinting of mitochondrial DNA? It is little tangential to the theme of your talk, but wants to know whether there's evidence for any imprinting of mitochondrial DNA in bees or probably any other system.
In general, no. As I've indicated, that I know nothing about bees other than the data I'm able to get out of the literature. In the other groups that I work on, which are mostly mammals, there's no evidence of imprinting.
So, short answer, no. Yeah. Any questions? Anything else? Well, if not, thank you so much, Steve, for waking up at the unearthly hour. And, we hope to see you again later today.
I hope I will be awake then. Thank you.
Well, we'll put you to sleep. Editorial board meetings drone on and on here just to extend your metaphor. So, anyway, so hope to see you there. So we can say that you joined from afar. Right? So, okay. So, Steve, you can go back to sleep. Yeah, and there are just a few hygiene matters that I have to deal with over here before we close the mini-symposium. So, first of all, I've been asked by Arun to tell all the students to collect their participation certificates and then go for lunch. Don't leave it for later because the person who has them also has to have lunch. So they can't be just waiting for you to come. So that's one. The other is, once again, I thank all of you for coming. It's good. I don't think Steve himself got the idea of how many people are in this room, but all the rest of us who spoke here and interacted with you, we are happy to see that big enough audience and so receptive. I think, and bear with me for keeping so obsessively to the time, because I think if we hadn't done that, then it would have been unfair to the last speaker who had probably woken up much earlier to participate in the talk. And actually, if anybody has any questions, that come up later, right, in the course of time, do feel free to write to the Journal of Genetics editorial office, and they forward the question to each of the relevant speakers, right? And so you don't have to feel that this is the last chance you get to interact with people. I think for many of you, it's probably the first formal talk on ChatGPT and to know how words can be found similar based on cosines and so on. So it's very fascinating. A lot to think about. Next time we'll have pomegranates. You know that it's good for you. And certainly on bees, it's inverted the prevailing notion. People might wonder why it is significant, but actually one of the big crises happening around the world, including in India, is that bee populations are dropping, right? Because of pesticide use and so on. And so there is a big concern about pollination, and all the multifarious ways that we take bees for granted. And people are really concerned about it. So it goes beyond just knowing where they evolved and so on, but also how well are we going to be able to conserve them in the future. So with that, I will, is there anything else that you have to say? Right. So I bring this meeting to a close. Yeah, thank you very much, and please join us for lunch.